When we speak of controlling microorganisms, what we mean is to effectively eliminate them within a particular environment. Controlling microorganisms require processes that remove or kill any viable microorganisms. Methods of controlling microorganisms can also entail suppressing any future growth and proliferation of microorganisms. The specific environment in which the process of controlling microorganisms is exerted can be the interior of a room, a solid surface, a given volume of liquid or gas, or even the living body of a human or an animal. Before proceeding to describe individual methods of controlling microorganisms in detail, it is essential to first define some key concepts. Disinfection describes a process whereby most or all viable microorganisms within a given environment are eliminated. However, disinfection methods are typically less efficient at eliminating microbial spores. Sterilization, on the other hand, describes a process whereby all viable microorganisms, including spores, are eliminated. Note that sterilization may still be ineffective at eliminating other undesirable biological agents such as prions and microbial toxins. Antimicrobial chemotherapy is a process that differs from disinfection and sterilization in that it specifically employs antimicrobial chemical compounds aimed at eliminating or suppressing a specific set of undesirable microorganisms within the living body of a human or other animal. Antimicrobial chemotherapy includes the therapeutic use of antibiotics, antifungals, antivirals, and so on. There are a number of factors to take into account when choosing the appropriate method for disinfection or sterilization. The first factor to consider is the physical state of the medium you are intending to either disinfect or sterilize. Is it a gas, liquid, or solid? If it is a solid medium, do you need to disinfect or sterilize just the surface of the solid or the interior of the solid as well? Next, you need to consider the physical properties of the medium. Is it sensitive to heat, pressure, or radiation? Likewise, the chemical properties of the medium must be considered. For example, if the medium must retain its specific chemical composition, this would preclude the use of chemical control agents. You may also wish to avoid undesirable chemical reactions such as covalent modifications or oxidations. If your medium possesses some form of biological activity, such as living tissue or viable seeds, you will need to use a control method that specifically eliminates microorganisms without significantly affecting the viability or biological activity of the medium. Finally, you will need to consider the cost of different control methods and whether a more efficient control method is justified by potentially higher cost. There is a wide spectrum of methods currently available for controlling microorganisms. These methods are commonly divided into larger categories. Physical control methods include the use of heat, pressure, and mutagenic radiation. Mechanical control methods consist of using physical filters to exclude or remove microorganisms. Chemical control methods include the use of chemical agents with antimicrobial properties such as surfactants, solvents, strong oxidants, alkylating agents, and biochemical inhibitors. Biological control methods, which will not be covered further in this lecture, include the use of organisms that prey on microorganisms, introduction of microorganisms that antagonize undesirable microorganisms, introduction of viruses that target undesirable microorganisms, or the introduction of enzymes or biological toxins with antimicrobial activity. Heat is perhaps the most commonly used physical method to control microorganisms. There is a wide spectrum of thermal control methods that differ in the way in which heat is transferred to the medium, the temperature range used, and the typical exposure time. Before taking a closer look at each individual thermal method of controlling microorganisms, it is useful to first look at how temperature tolerance in microorganisms is quantified. The most widely used metric to describe temperature tolerance in microorganisms is the so-called decimal reduction time, or d-value. The d-value is always given for a particular temperature, shown here as subscript capital T. The decimal reduction time at a given temperature is defined as the time required to reduce the number of viable cells of a specific microorganism by 1 order of magnitude, i.e. a factor of 10, which is the same as a 90% reduction in viable cells. This means that exposing a specific microorganism to the same temperature as indicated by the subscript of the d-value for a period of time that equals the decimal reduction time, the number of viable cells will decrease by a factor of 10, i.e. a 90% reduction. If the exposure time is twice that of the decimal reduction time, the number of viable cells will decrease by a factor of 100, i.e. a 99% reduction. If the exposure time is 3 times that of the decimal reduction time, the number of viable cells will decrease by a factor of 1,000, i.e. a 99.9% reduction, and so on. How is the decimal reduction time determined for a specific microorganism? This can be done quite easily by exposing the microorganism to the temperature in question and then measuring how cell viability decreases over time. Imagine that we have a population of 1 million viable cells of the microorganism we are interested in. We expose this population to a set temperature for 6 seconds and observe that the number of viable cells has dropped to 1,000. Note that the y-axis has a logarithmic scale. Using this formula, we can calculate the decimal reduction time for this particular temperature. We first enter the values for our 2 data points. Note that I'm expressing the number of viable cells in base 10 for clarity. By taking the base 10 logarithm of the viable cell numbers, we now get these values. We then simply do the subtractions to get the numerator and denominator. After dividing the numerator by the denominator, we get a decimal reduction time of 2 seconds. We can further illustrate this by plotting a line through the 2 data points, again noting that the y-axis has a logarithmic scale. Now we can clearly see that a 2-second exposure time corresponds to a 10-fold reduction in viable cells from 1 million to 100,000. Another 2 seconds of exposure corresponds to yet one more 10-fold reduction in the number of viable cells, this time from 100,000 to 10,000. Yet one more 2-second exposure corresponds to another 10-fold reduction in the number of viable cells, from 10,000 to 1,000. We will now take a more detailed look at the spectrum of thermal methods that can be used to control microorganisms. Incineration, red heat, and flaming are 3 simple, high-temperature methods to eliminate microorganisms. Incineration involves the complete combustion of the medium at high temperature and is therefore only really applicable to the safe disposal of biohazardous waste. Sterilization by red heat or flaming involves the use of a Bunsen burner, and both methods are used during routine laboratory work involving heat-resistant metal instruments and glassware. Red heat sterilization involves placing heat-resistant metal instruments in the flame of a Bunsen burner until the metal glows red hot. The inoculation loop seen here is made of highly heat-resistant metals such as tungsten or platinum and will therefore not melt. Flaming is primarily used to rapidly sterilize metal tools and the openings of glass containers such as flasks and test tubes. Since flaming is only used for a section of the object, it should be thought of as a temporary sterilization method that only reduces the chance of contamination but does not eliminate it. Hot air sterilization is a simple way to sterilize heat-resistant solid materials such as metals, glassware, ceramics, and textiles. Since heat transfer is accomplished through conduction, hot air sterilization requires more time than other thermal methods. The temperature maximum determines how long the process should be with lower temperatures requiring longer incubation times. Some standard sterilization setups are shown on this slide. We now move on to thermal methods that use moist heat transfer. Boiling is perhaps the simplest method of moist heat sterilization, but is rarely used today except in cases where there is no access to more modern heat sterilization equipment. Boiling works well for sterilization of heat-resistant liquids with boiling points at or above 100 degrees centigrade. Boiling can also be used to sterilize heat-resistant solids by, for example, immersing them in boiling liquid, provided the solid medium will not react chemically with the water or dissolve in it. Boiling is effective at eliminating vegetative cells of most microorganisms as well as viruses. However, boiling is less effective at eliminating microbial spores as well as non-cellular, undesirable biological agents such as prions and microbial toxins. Boiling will also not be effective at eliminating hyperthermophilic archaea, although this group of microorganisms is rarely a concern for most applications. Tyndallization, named after the Irish physicist John Tyndall, is a sterilization process that uses sequential boiling steps to better eliminate microbial spores. Tyndallization of liquid media involves boiling the medium for at least 15 minutes, which will kill vegetative microbial cells. The medium is then allowed to cool down for 24 hours, during which time some spores will germinate and become vegetative cells. The medium is then boiled again, which will kill any new vegetative cells. This is followed by another 24-hour cooling down period where remaining spores may germinate. The medium is then boiled a third and last time to eliminate any new vegetative cells. Tyndallization of solids uses a similar procedure. The solid medium is exposed to hot steam for 30 minutes, which kills vegetative cells. The medium is then allowed to cool down for 24 hours, which allows spores to germinate and become vegetative cells. Any new vegetative cells are then killed by another exposure to hot steam for 30 minutes. These steps are then repeated as before. As with boiling, Tyndallization is rarely used today, except where there is no access to modern heat sterilization equipment. Pasteurization, named after the French microbiologist Louis Pasteur, is a thermal process for reducing the number of viable microbial cells in beverages such as milk, beer, and fruit juices, and thereby extend their shelf life. The goal of pasteurization is to reduce the population of viable microorganisms in the liquid medium as much as possible without significantly affecting properties such as taste and structure. There are several variants of pasteurization with regards to temperature and exposure time. Typically, a lower temperature will require a longer exposure time. One exception is thermization, which is sometimes used for milk that will be used for making cheese. In this particular case, there are several downstream process steps in cheesemaking, such as lactic acid fermentation and salting, that will serve as additional controls on the growth of undesirable microorganisms. Autoclaving is a widely used sterilization method in both industry and clinical settings. Autoclaving uses pressurized superheated steam for heat transfer, which is much more efficient than hot air and therefore requires shorter process times. The standard protocol for autoclaving is 121 degrees centigrade for at least 15 minutes. Autoclaving is suitable for sterilization of solid and liquid media, provided they are both heat and water resistant. Autoclaving is effective at eliminating all types of microorganisms with the exception of hyperthermophilic archaea. However, this group of microorganisms is rarely a concern for most applications. It is worth noting that autoclaving is not effective at eliminating prions or heat-tolerant microbial toxins. Radiation-based thermal methods such as infrared radiation is useful for rapid heat disinfection or sterilization. Infrared radiation specifically is well suited for sterilization of solid surfaces. Microwave-induced heating for the control of microorganisms is an emerging technology in food processing where it can extend the shelf life of packaged pre-cooked meals and salads. An alternative to thermal control methods is the use of high-pressure processing, so-called pascalization. This method uses high hydrostatic pressures that can exceed 1,000 times the atmospheric pressure at sea level. At these pressures, both protein and membrane structures are disrupted, leading to the death of undesirable microorganisms. At the moment, this method is primarily used in food processing. Mutagenic radiation is an effective method for sterilization and includes ultraviolet radiation as well as ionizing radiation. Ultraviolet radiation leads to the formation of pyrimidine dimers in nucleic acids such as DNA and RNA. Therefore, the use of ultraviolet radiation is only suitable for inanimate materials, particularly solid surfaces, although it can also be used for purification of air and water. Ionizing radiation, such as X-rays as well as alpha, beta and gamma radiation, can fracture the backbone of nucleic acids. X-rays and gamma radiation have the added benefit of being able to penetrate solid materials, which allows for the sterilization of solid interiors. Filtration is a mechanical method of eliminating microorganisms by passing the media through a perforated barrier, the filter itself, which excludes particles exceeding a certain size. Most filters used for sterilization consist of polymeric materials that form a mesh-like structure. Common filter materials include nylon, nitrocellulose, and glass fiber. Filtration is suited for the sterilization of gases and liquids. The average pore size of the filter determines how efficiently it can remove microorganisms. Filters used for sterilization require an average pore size of 0.22 micrometers or smaller. Such filters will effectively remove all cellular microorganisms as well as larger viruses. However, this average pore size will not be sufficient for the removal of smaller viruses, prions, or microbial toxins. These agents require filters with smaller pore sizes, so-called nanofilters. A widely used filter standard worth mentioning is the high-efficiency particulate air filters, which are used extensively in industry and clinical settings. The last group of control methods to be discussed in this presentation are the chemical methods. Chemical agents that are used in the control of microorganisms can be divided into a number of functional categories depending on how they are used. Sanitizers are chemical agents primarily used for cleaning that also have moderate antimicrobial activity. Disinfectants are chemical agents able to kill vegetative cells of microorganisms but are less efficient at killing microbial spores. Disinfectants are intended for inanimate surfaces and liquids. Antiseptics, on the other hand, are meant for use on living tissue such as skin and wounds. The purpose of antiseptics is specifically to control pathogenic microorganisms. Antimicrobial chemotherapeutic agents are also used in order to control pathogenic microorganisms. Unlike antiseptics, these agents are used within the living body of a human or other animal and include antibiotics, antifungals, antivirals, and so on. This category of chemical agents will be discussed further in the second part of this lecture. Chemical pesticides are antimicrobial chemical agents that are used to control undesirable microorganisms in agriculture and forestry. Chemical preservatives are antimicrobial chemical agents used to suppress microbial spoilage. This category of antimicrobial chemical agents will be discussed further in another lecture. Note that some chemical compounds can belong to more than one category. It is also possible to classify antimicrobial chemical agents according to their efficacy. Microbicidal chemical agents control microorganisms by killing them. Microbiostatic or microstatic chemical agents control microorganisms by suppressing their growth. In many cases, the efficacy of a particular antimicrobial chemical compound depends on its concentration. Antimicrobial chemical agents can be used in different formulations depending on the properties of the individual compound. The choice of which type of formulation to use depends a lot on the properties of the medium to be treated, as discussed in the beginning of this lecture. Surfactants exert their antimicrobial activity by disrupting membrane and protein structures. These compounds can be used both as disinfectants and antiseptics. Alcohols work much the same way as surfactants by disrupting membrane and protein structures. Alkylating agents are highly reactive compounds that covalently modify biological molecules, which make them highly toxic, and they can therefore not be used as antiseptics. Ozone and peroxides both contain unstable oxygen-to-oxygen single bonds. As these compounds decompose, they produce highly reactive oxygen species. Halogen compounds, especially iodine and chlorine, have good antimicrobial properties due to their high electron affinity, which make them strong oxidizers. D-block metals such as copper, zinc, silver, manganese, cadmium and mercury have antimicrobial activity at elevated concentrations. The precise mechanism of action differs. D-block metal ions can disrupt metal homeostasis in microbial cells, for example by displacing other metal ions within proteins or inhibiting the uptake of essential trace elements. Some D-block metals can act as strong oxidants. The final group of antimicrobial agents are notable by their ability to selectively inhibit particular biochemical processes within the microbial cell. This group of chemical compounds is extremely diverse, and only 2 examples will be given here. Azoxystrobin is a broad-range fungicide that specifically targets the respiratory electron transport chain. Triclosan is an antibacterial compound that is sometimes used in soap. Triclosan specifically targets bacterial fatty acid synthesis.